Okay, so hi everybody. Thank you for being here this early in the morning for I think the last Macroevolution session. I'm Amanda Doyle. I'm a PhD student in David Jablonski's lab at the University of Chicago, and today I'll be talking about a project I have on genus level patterns in bivalve body size evolution. So before I begin, I'd like to thank all these people for their help in this project and the presentation, as well as these funding sources for their money. That's nice. So, getting into the science, one key feature of understanding evolution is determining how different evolutionary processes can produce different distribution shapes. So here's a figure from McShay's 1994 paper in which he introduces the ideas of passive and driven trends, which essentially describe different means by which clades can attain some mean trait value. While not a foolproof way to differentiate between these trends, skewness can help to indicate whether a passive trend, which tends to produce a skewed distribution, or a driven trend, which tends to produce a symmetric distribution, is present. And the direction of skew certainly helps to determine where there may be some boundary and where a clade can move in terms of its mean trait value. So a fantastic clade to use as a study organism for such studies are the bivalves. So they're incredibly taxonomically diverse, both now and in the geologic past. They have a very long and a very good fossil record, and they're highly variable in numerous traits. One particularly useful example for this type of study is body size, with bivalves sporting sizes anywhere from under a millimeter to over a meter. Bivalve body size can be characterized in a number of ways, but here I'm referring to the geometric mean of their length and height. This is a metric that correlates well with many other ways to calculate their body size. So size is a particularly useful trait to use when studying evolutionary trends in bivalves because it's not only variable across the clade, but it also shows a strong signal of differentiation across different biogeographic regions. In previous work that I've presented, I've found that modern bivalves differ strongly in their body size by hemisphere, with northern hemisphere bivalve faunas consistently sporting larger bodied faunas than in the southern hemisphere. This contrast also increases in strength as you move poleward and holds even for endemic bivalves alone. However, there are some indications that this really strong interhemispheric contrast is a relatively recent phenomenon. I've found that at least at the poles, there's no contrast in body size between the northern and southern hemisphere faunas at the Paleocene, while there's a strongly significant contrast in these faunas today. Moving back to the present day, it's also apparent that the relationship between body size and skewness has some nuance to it. So here I show a plot that I first presented last autumn, in which each point is the median body size of each bivalve with at least 10 extant species, plotted against the skewness of those distributions of sizes. So as you can see, while the overall pattern shows little general trend, there is clearly a difference between the smaller bodied and larger bodied genera, with the former showing largely symmetric distributions, while the larger bodied genera also frequently have left skewed distributions. Notably, when these genus-level body sizes are split into equal tertiles, representing three size classes, that is small, medium, and large-bodied genera, there's no distinction between the skewness of the medium and large-bodied genera. But both of those large size classes do differ strongly from the skewness of small-bodied genera. So these contrasts in body size by biogeographic region and skewness and the apparent Cenozoic onset of this interhemispheric body size differentiation raise many questions about how these patterns may have been achieved. So the question that I'm interested in investigating is how patterns in genus-level body size evolution differ by biogeographic region and by body size class. In order to investigate this, I sought and continue to seek to test how body sizes have changed between the first known fossil of each speciose bivalve genus and in conjunction with skewness of their modern distribution, how the prevalence of driven, passive, and a lack of trend in body size evolution can be contrasted by their body size class today and the biogeographic region that each genus is now most dominant in. Given the prevalence of small-bodied bivalves in the southern hemisphere and the fact that small-bodied genera tend to produce mostly symmetric distributions in body size, I expected that most small-bodied genera would show a mix of no contrast in their first known fossil and current median, and driven trends in which the earliest fossil member is significantly larger than the modern genus median. That is, their body size has decreased since the origin of the genus. In contrast, given the prevalence of large-bodied bivalves in the tropics and the northern hemisphere, and the fact that large-bodied genera tend to sport symmetric and left-skewed distributions in near equal frequency, I expected that most large-bodied genera would show a mix of no contrast in their first known fossil size and current median, and passive trends in which the earliest fossil member of each genus is significantly smaller than the modern genus median. Thus, body size has increased since the origin of the genus. In order to test these hypotheses, I used the Jablonski Lab's massive modern bivalve occurrence data set and pared it down to genera with at least 10 extant species. I also used a data set of body sizes with the first known fossil occurrence of each of these genera, which I'm still collecting data for. Currently, I have sizes of 75 genera with at least 10 extant species. Then the directions of body size evolution were tested using Mann-Whitney use tests with a Bonferroni correction here, but I'm currently working on simulation studies for a more refined analysis of these data too. I should note that unlike previous analyses that I've presented on skewness, I split genera here into just two body size classes, small and large, because like I mentioned earlier, distinguishing between medium and large bodied genera in terms of characteristics of their distribution is basically not terribly useful. The cutoff for a small-bodied genus was anything with a median size under 15.44 millimeters. This corresponds with, and indeed is partly defined using, that sharp contrast in the skewness of the distributions. This cutoff leaves roughly 2/3 of the genera in the large body size class and 1/3 in the small size class. So each genus's body size distribution consisted of the maximum body size of each extant species in that genus. Here I have the extant distributions of 3 genera, along with red X's denoting the body size of the first known fossil of each of these genera. So the data show numerous patterns of fossils that are completely outside of their modern distributions, like Sphenia on your left here. There's also some examples of genera that fall roughly on the median of their current size distribution, like Cyclocardia in the middle. And then there are, of course, genera that fall within their distribution, but towards one of the tail ends of it, like Limopsis on the right. And I should note that these fossils can fall on either the left or right side of these distributions. There wasn't really any strict partitioning against one of those. So moving on to results, here I have plotted the modern genus median sizes against the modern distribution skewness, with each point colored by how many interquartile ranges away from the modern median the first fossil size is. So fossils that are smaller than their modern median are in some shade of blue, and those that are larger than their modern median are in some shade of red. You'll see 2 black dots here. These ones are extreme outliers. They are far smaller than their modern medians. They're upwards of 10 IQRs away, while you can see that usually these fall between -4 and +3 or so IQRs away. So there also seems to be some partitioning here of where these increases versus decreases in size can be based on size and skewness. You'll note that these 3 red dots seem to be clustered on the small body size class, and while at least the darker blue dots tend to mostly be in the large body size class. And furthermore, the red dots are well within the symmetric or even right-skewed sections of the plot, while the darker blue dots largely tend to be symmetric or even strongly left-skewed in their distribution shape. However, when tested for statistical significance, the majority of genera tested here sported no significant contrast between the body size of their oldest member and the median of the modern distribution, with increases in body size being the second most common trend. A mere 4 of the 75 total genera actually appear to have significantly decreased in their median size since their origination. Also surprisingly, small and large-bodied genera tested here don't significantly differ in their direction of change, although I should note that these comparisons can really only be performed statistically for genera that either increase in size or have no change in size. 4 is a difficult number to do statistics with. There's also no significant difference in the skewness of a direction of body size change, though this comparison would also be made far more robust if genera that decreased in size could be included, as that would indicate that passive and driven trends are equally as common in genera that have increased and decreased in their body size. So finally, here I show the number of genera of each of these 3 body size change categories. So an increase in their size through time, no change in size through time, and a decrease in size through time. that are now most speciose in different biogeographic regions. So from left to right, these regions are the southern hemisphere high latitudes alone, southern hemisphere high latitudes and tropics, the tropics, northern hemisphere high latitudes and tropics, northern hemisphere high latitudes alone, and the whole globe that is truly cosmopolitan taxa. This is a bit of a complicated plot, so first we'll focus on genera that show a significant increase in their body size since their origination. As you can see, the region most of these genera are most prevalent in is the tropics, with a significant chunk of these genera most common in the southern hemisphere high latitudes and in the northern hemisphere tropics and high latitudes as well. If we move on to the genera that show no significant change in size, we again see that the tropics is the most common region that these genera are most speciose in. And again, we see that the southern hemisphere high latitudes and northern hemisphere tropics and high latitudes are the second and third most speciose regions for these genera. Given that the number of genera exhibiting no change in median body size is slightly less than 2.5 times that of genera showing a significant increase, we can clearly see that these contrasts in the direction of body size evolution are found across numerous biogeographic regions with little difference in their prevalence by region, at least between these two size change categories. This seems to indicate that despite strong contrasts in skew by body size class and body size by region, there may be more homogeneity in the evolution than in these modern patterns that those evolutionary histories have produced. In conclusion, these 75 speciose bivalve genera tend to show largely no net trend in their body size evolution, with a substantial number of genera showing a net increase in body size and a mere 4 showing a net decrease in their body size. Clearly, despite the myriad of modern contrasts in the fauna, speciose bivalve genera seem to sport no change, passive trends, and driven trends in their body size evolution across both small and large-bodied genera, although the lack of data on genera that have truly decreased in their body size through time makes an overall conclusion difficult to pinpoint. Though there's clearly little difference in the prevalence of these trends for genera that have increased or have no net change in body size, it's possible that genera that have decreased in time through their evolutionary history will have some distinct patterns. At least between genera that either got larger or had no net change in size, broadly similar regions of the globe are occupied by both groups, though not much can be said as it stands for bivalve genera that have decreased in their body size. Finally, it should be said that this project still has a lot of work left to it, in a good way. Not only are there more speciose genera to include, but I have simulation studies to complete that may help shed light on the finer details of how these distributions could arise and which are truly strong deviations from their first known fossil. Thank you all for listening. I'd be happy to answer any questions that you may have. So that's a great question. As far as the age of the clade and the skewness, not really. I'm looking more into whether there might be some signal for the age of the clade and at least the direction of their size change. In fact, if we go back to my hypotheses, I didn't get into this because it takes a little bit more time, but part of the reason that I think that there could be this partitioning of whether there's no net change or a driven trend, say, in the small body bivalves depends on whether they're quite young and thus have, originated in a fairly cool kind of climate, or if they're quite old and presumably have seen, the really intense warm periods of like the PETM or late Cretaceous and such. Cool. Okay, well, it's already up, so with that, thank you guys.
Thanks everyone for coming. I'll do my best to stand in front of the microphones. I have a habit of moving around a lot. So I'm Ian, I'm visiting from the Natural History Museum in London. Oh, is it? Oh, I have to wait for the bell. Sorry, I was going by the clock, my fault. I'll start again. Thank you. I'll be somebody else this time. Go. Is that the one? Ah, yeah. It probably said that in an email somewhere that I didn't read. Cool. Thanks for coming along. I'm Ian. I'm going to be talking about the tempo and mode of morphological evolution in a group of Australian skinks. This is a project that is mostly wrapped up now, so there's a preprint online. You can scan the QR code. You can decide at the end if you'd prefer, because there's another QR code at on the very last slide. So let's get started. Some way. Oh. Haha, sorry about that. Cool. So the basic argument I'm going to try and convince you is that the morphological novelty in this group of Australian skinks has evolved via discontinuous processes, so via evolutionary bursts in morphology. And skink phylogenetics and taxonomy are a bit of a mess because they're really rapid radiation, so we don't have a great handle on them. But we do understand this individual group quite well. So we're going to talk about the Taliquini, which is a tribe that's native to Australia, New Guinea, and the Solomon Islands. There's about 60 species, about 9 living genera. And they're an interesting group to ask these kinds of questions about morphological evolution because they're really varied in their morphologies. So on the left side of the slide, in the middle, you have a really kind of traditional looking skink. 4 well-developed limbs, overlapping scales, long tail. Above it, you have something that's really kind of outlandish for skinks. This is Tribolinotus. It's called the crocodile skink. Has this really kind of goth makeup going on. On the top right, you have Cyclodomorphus, which looks like you took a normal lizard and stretched it like silly putty. And then you have a diversity of other incredible forms, including the shingleback lizard in the middle on the right side, which I'm going to talk about a bit more. First step in any of these kind of studies is we need a reliable tree. So we use an exon capture approach using anchored hybrid enrichment to build the tree for this clade of what we call social skinks. Call them social skinks because they often lived in family units. I'm not going to cover that today, but they're a very cool group. But what's important to identify is that the Australian species form a single clade. That's the majority of the species richness, which is a nice group to kind of contain and study. We used a combined evidence approach to date the tree using molecular data and morphology. You can read a bit more about that in the paper if you're interested. But I'm going to show you a little bit of the diversity that's in the group because I think this is what's cool and why they're an interesting group to study. We have small little things with spiny tails that live under tree bark or in rock crevices. This is a Gurnia depressa. We have the heaviest skinks in the world, so blue-tongued lizards that get really big and wander around the open spaces of Australia. They're kind of like Australia's tortoises. And then we have the biggest skinks in the world, a thing called Carusia zebrata, the prehensile-tailed skink or monkey-tailed skink. It's an arboreal species that lives in the Solomon Islands. So you have a lot of ecomorphological variation going on here. But how does this variation evolve? How do you get from a standard little thing to something quite exceptional? How do we go from this little traditional skink to a monster like the shingleback lizard? Or there's recently described a thing called Taliqua frangens, which was a shingleback, but even bigger, maybe a couple kilos in weight. Traditional kind of evolutionary models like Brownian motion will say that we're exhibiting a gradual change through time. But I'm going to kind of put forward some alternative models that proportion the evolutionary change into a heterogeneous kind of process. So we have kind of fits and starts in morphological evolution. We need a data set, so we went out and we collected 21 linear measurements from, I don't know how many I said, 650 specimens across all of the species that are inside in this group. And we processed the data in pretty standard ways where we brought measurements down to species means, and then we removed the effective size by using log shape ratios, and we retained size as a separate variable. Then finally, we analyzed traits individually, but we also fit them into modules. So we fit head, body, limbs, and tail into separate multivariate morphological modules. And so when we think about the evolution of these individual traits or the modules, we can kind of think about the accumulation of variation happening linearly, like under Brownian motion, or under an early burst scenario where we accumulate a lot of variation fast and then it kind of tapers off, or this last one, which is what I'm going to focus on for the rest of the talk, this variable rates model in Bayes traits, which gives us these kind of jumps in trait evolution. So when we take the individual traits, we run them through these series of models like Brownian motion, early burst, OU, and this variable rates model, we get strong support for most of the traits for variable rates, so a heterogeneous evolutionary process. One thing to note is you'll see that interlimb is one of the traits, and it's highlighted in yellow. It's supported by a jump process, which is conceptually similar to this variable rates process. So I'm going to talk about that trait specifically in a minute. So how does evolutionary change kind of split up across the tree when we use this model? Well, we estimate individual rates for each branch of the tree. I like to think of kind of an analogy is that if we're doing divergence dating, you allow, in a relaxed clock method, you allow individual branches to have different substitution rates. It's conceptually similar to that in that we're allowing a different evolutionary rate for each branch of the tree. We then use, so here we have got kind of the background rates in like light yellowy or whatever, and the faster rates in blue. And then Bayes traits uses a reverse jump algorithm to identify branches that deviate in their rate from a background rate. So these are significant shifts in evolutionary rate. And you'll see a bunch of those shifts are in a clade, this cyclodomorphous and taliqua clade. So this is the blue-tongued lizards. And you might not know anything about them, but I'll just tell you, they've got, we're looking at just a single body module here, a single module, which is the body. And these skinks have gotten really long trunks, like long torsos, and then really, some of them have gotten really wide and deep bodies as well. So there's a lot of variation happening within that individual group. And we can see the branch with the highest rate is about 15-fold faster than the background evolutionary rate, and that's happening on the branch that leads to that taliqua and cyclodomorphous clade. And we can visualize that on an individual trait in interlimb length here, where we see a huge jump in the evolution of that trait from its ancestral state to the ancestor of taliqua and cyclodomorphous. So a lot of change happening on an individual branch. So I'm going to give you my kind of 3 takeaways here to think about, and then I'm going to give you one fun example to end on. So the things we want to take away here are that the tempo and mode is kind of this punctuated gradualism, where we have a gradual background rate of evolution that's happening, punctuated by bursts in evolutionary rate that result in big changes. These shifts, or these bursts, are actually quite uncommon. 0.003 to 0.005 or 0.05 shifts per million years. So they're not happening that frequently. And lastly, these bursts or shifts are can be nested to generate new morphologies. And this is particularly interesting because they can either be staggered through time, or they can be concurrent, so they can happen at the same time or on a similar branch. And I'm going to give you an example of how we can visualize this, how these nested shifts contribute to new morphologies. So we're going to build a shingleback lizard. So shingleback lizard's a really cool animal. It's got this massive armor on its back, osteoderms set in the scales. They're kind of like a giant chainmail. They live a really long time. They're monogamous, and they wander kind of open habitats in Australia. And they're one of the more extreme morphologies in the group. So we're going to go through the tree from that red dot at the common ancestor of the group to that purple point. Our first shift happens is a kind of concurrent shift in the body and limb proportions. Limbs get much, much shorter, and body gets lengthened and widened. So those things kind of happen at the same time, somewhere at the ancestor of taliqua and cyclodomorphous. Our next shift is in the head proportions. We get a widening and a deepening of the head, followed by a big jump in size that leads to the genus Taliqua, which these are big lizards compared to what you think of as a general skink being. It's followed by a shift in the tail, where to get to that shingleback stumpy tail, which kind of imitates their head, you really need to shorten up the proportions. It's a wider, shorter tail. And then finally we get to our Taliqua rugosa, the shingleback. So these staggered shifts lead to these really new, incredible morphologies. So I'm going to head you off with a few questions that you might have or you might be thinking, is this not just punctuated equilibrium? Well, we don't have evidence that these shifts are happening at cladogenetic events, and we're suggesting that there's kind of gradual change happening along the branches instead of kind of stasis happening along them. And then this is a question that I think is particularly interesting, and you might too, is, what's the role of selection happening here? Obviously, if we say that these branches are undergoing selection, we can remove the need for an increased evolutionary rate. We can have a kind of a directed walk that takes us from our ancestral phenotype to our new morphology. And that's already been implemented in a different model in Bayes traits that's called the fabric model, which we didn't use here, but is conceptually pretty cool. That's it for me. I just want to thank co-authors on this work, but more importantly, funding. So I got support from Mary Curie Fellowship to host me at the Natural History Museum. Thank you very much to Natalie Cooper and her macro group at the NHM, and ANU for previous support. You can scan the QR code if you want to read a bit more about these really cool lizards. Thanks for coming. Yeah, go ahead.
Great job, thank you. I'm wondering if you've looked at the nature of the phenotypic change in relation to the reverse. So I'm thinking about your interlimb length, for example.
Yeah, that's a great question. I don't know. There's actually quite a bit of osteology work on these things. I haven't looked into that, but that's a great idea. We know in snakes that's usually what happens. When you get really long constrictors or whatever, they just chuck in more vertebrae. But I'm not sure what's happening in these. I didn't think about that, but I'm going to look into it after this. Thanks. Go ahead. Cheers.
I was just wondering, so that we first seen evolution along the lines of blue tongues.
Yeah.
It's also a long branch.
Yeah.
Tree. So I was wondering what your thoughts on possible extinction along that branch is, or just generally the role of extinction.
Yeah, that's a totally valid question. We had that from reviewers as well. We did this combined evidence analysis where we put in as many fossils as we can. So there's 5 or 6 good fossils that we can fit into the tree. It doesn't qualitatively change our results, but there's, if there's 60 species, there's 60 extinct species too, probably. So it's hard to answer that question, but I think the way that the really unique morphologies are nested deep within the phylogeny suggests that you had to get to these dramatic changes relatively rapidly. The branch that leads to Taliqua is actually quite short, so I don't know what I do here. Leave it? Cool. Okay. That's good advice generally. Thanks, everyone.
Welcome, everybody. My name is Jake Berv. I'm a postdoc at the University of Michigan. Recently started a new job, well, actually about a year ago, more focused on AI. And pretty much this is actually the first talk in an evolution meeting that I've given in over 10 years that's not about genetic data. So congratulations to me involved in this. Okay, so yeah, as I said, we're currently in the middle, it's sort of deep in this revolution of genomic data. I apologize for the tiny aspect ratio, but so if you can't see in the back, please feel free to step forward and take a better look. So we're currently kind of deep into this genetic data revolution, and I started my career right before this happened, and so most of my research interests were sort of dictated by this data, this transformation in our ability to collect genetic data. And the cost of generating sequence data, as we all know, has really dropped enormously, by like 100,000-fold in the last 15 years. And this is partly due to new compute capacity to actually process data coming off these sequences, and also chemistry, revolution in the actual sequencing chemistry. So we're deep into this genomic data revolution. And even though we've made tremendous progress on generating morphological data to accompany these beautiful genomic data sets we've generated, it's still kind of lagging, and we're kind of just getting to this point where we're able to generate large morphological phenotype data sets to complement the genetic data sets that we're not generating. So typically when we generate morphological data sets, it really just requires manual labor, as everyone who does this knows. So the main cost of doing this is the cost of the time and the cost of the labor. So I study birds mostly, and so we typically use digital calipers, and we actually look at a specimen in a physical collection and measure different aspects of it. And there was a really wonderful paper published a couple of years ago that published this data set called the AvoNet data set that curated a very, very large avian functional trait data set. And this is basically looking at gross anatomical proportions of the external phenotype. It includes over 90,000 specimens. Over 150 researchers contributed to this, but it took decades to generate this. And so even though this is a fabulous step forward, we need to go faster if we're going to understand biodiversity while we still can, and I think we can. So enter AI into this equation. So let's think about bird skeletons for a second. So bird skeletons are complex geometric objects, and it wasn't at all apparent to me that we could actually do this when I started working on this about a year ago. Can we use AI to measure bird skeletons and extract useful data? Well, it turns out we can, and this is basically a solved problem now, especially now that we're sort of in this AI revolution. There was a paper published by my current advisors about a year ago that describes a neural network approach for basically measuring bird skeletons, specifically passerine bird skeletons, songbird skeletons. And in the last year and a half, we've collected data for over 15,000 skeletons, really more about 20,000 skeletons now. It's taken less than 2 years and less than 10 people. So even though the data set is currently not quite as large as the AvoNet data set, the rate at which we're generating data is dramatically faster. And so in the next few years, we expect to dramatically overshoot what the scale of the AvoNet data set. So this is how it works. We have a very cheap and simple imaging setup. The cameras are pretty high quality, but the overall cost is only a few $1,000s to build this whole thing. It's just a simple platform with a camera and some light sources. You take one two-dimensional photograph, and the whole process takes about 45 seconds. That was sort of a restriction that we had. In order to go through whole collections, it has to be super, super high throughput, super fast. So 45 seconds to go from the specimen on the shelf, on the platform, take the picture, and back in the box, and then you're done. And you take an image, and the AI sorts it out, basically. I'm not going to talk really about the AI system at all, but the short version is that it works super well. The error is really low, about the same as human error. And if you want to read the paper that describes how the method works, I'm happy to send that to you. It's like magic. It actually does seem to work, at least for the sort of the gross proportions that we're targeting right now. And I'm just realizing there's no timer on here. Oh, no, there is a timer. Okay, cool. So here's the spatial distribution of the data currently. There's a heavy bias towards North American specimens because we started in the University of Michigan collections. So we've gone through the whole collection at UMMZ for passerines. And we're going into Chicago, and now we're expanding to the UK. So yeah, if you have a specimen collection of at least passerine skeletons for now, and you would like to collaborate with us on this initiative, please get in touch, and we'll try to set it up. We are eager to expand outward. We have nice sampling within and between species. Here is a diagram showing passerine songbirds, so sub-Aussians and Aussians, with bars just representing sort of the relative sampling. So we have nice sampling within species, which is nice, which is unusual to have. And we're hoping to come up with projects that will actually leverage the information we can extract from sort of good information about within and between species variation across the skeleton. Okay, that's all I'm going to say about the data set. I really have spent most of the last year working on new methods to like help us analyze this scale of data, and so that's what I'm going to talk about for the rest of the talk. So typically when we fit evolutionary models like Brownian motion, we're interested in estimating evolutionary parameters like this sort of sigma squared parameter, and we can fit these models with likelihood to evolutionary trees. In this case, I'm just showing a sort of a cartoon of an avian tree. But the key thing we're kind of interested in is this rate parameter, at least in the case of Brownian motion. This rate parameter kind of controls like the wiggliness of how the trait varies through time, and you can estimate that parameter from the data. We can extend this to multivariate data by conceptualizing sort of what's called a variance-covariance matrix, where the diagonal basically represents the individual trait variances, and the off-diagonals represent the trait covariances, so how the evolutionary covariances of the phenotype. So when you fit a model like this, it looks kind of like this. You can see it's like sort of a wave of traits going through time. Here's just a couple of little cartoons. Going through this quickly. But maybe you want to fit a more complex model, like a mixture model, where you have multiple variance-covariance regimes across the phylogeny. And that might look something like this. But as you can see, there are a lot of parameters in each of these regimes, and so actually figuring out where to place these is a challenge. And ideally, we would try to learn this process directly from the data, but as the tree gets bigger and bigger, the number of possible configurations of these, of how you can parameterize this is like infinite. And so you need a heuristic search some kind of efficient heuristic search to place these kinds of regimes. And there's been a lot of research on this over the last 15 years about how to do this for sort of smaller scale problems. But if you are able to do this, you might be able to ask, well, if your initial question is sort of how skeletons evolve, how does the phenotype evolve, and you can accept that maybe a similar question is how do the parameters evolve. Those may not be exactly the same question, but if you believe they are, you can start to ask questions like whether or not skeletons the evolution of the phenotype might be related to Earth history. So, for example, what do I mean by this? Like let's think about climate change over the past 100 million years. Can we look for changes in these parameters that are associated with major climate shifts? And we can potentially do this by looking at where these parameters might change on a phylogenetic tree. So here, let's go through this quickly. Okay, so when I started working on this, the scale of the data was crazy for this type of problem, and I kind of wanted to do like a no compromise kind of approach to this, because every kind of method that was out there required dramatic reductions of the data set or reductions of the phylogenetic context, and I didn't want to do that. So I built a new way to do this. So it's based on PCM-Fit, the algorithm from PCM-Fit, but it's using a much faster underlying machinery from MVMorph. I'm not going to go into exactly what the algorithm is doing. I can talk about that later. The name of this thing is going to be Bifrost, I think, for various reasons. I can get into that. But it has some really nice properties as a sort of hypothesis-generating machine. You don't need to do any kind of ordination. It works on hundreds of dimensions at the same time. It's super fast. And you run it with like a regression model. So you can naturally include like size or factor variables or whatever. You can come up with some, if you want to look at variable size allometry across your clade, you can account for that naturally in the model fitting without having to do any kind of pre-processing of the data. And it works with fossil trees, tip-dated trees, molecular trees. I programmed algorithms for looking for divergent and convergent patterns. I'm also building a simulation framework that allows anyone to take their own data and assess the performance of the method, the theoretical performance of the method for their particular data set, which should be useful. And it's currently only implemented in sort of a multivariate Brownian motion framework, but we hope to bring it to an OU framework eventually, extend it to that. How well does it actually work? So here's just an example of some simulations that I did last week, and still this is ongoing research. Where we're simulating 10 independently derived shifts in the phenotype variance-covariance matrix. And the model recovers almost all the shifts, but not between 9 and 10. It's like 90-something%. It's getting like 90-something% of the simulated shifts, at least in this context. So that's good news. It seems like it's working. So that's all I'm going to say about the model itself. And here's some actual empirical results. When we apply this approach to this giant phenotype data set that we generated for passerines. I should also thank Ian for giving us a nice introduction to basically what I'm going to talk about. Yeah, so we find that we can sort of firmly reject the null hypothesis that the process of evolutionary change has been homogeneous across Pasteurian evolution. We find many discrete jumps in the rate of phenotypic evolution. I'm just going to talk about rates today, not covariances, but ask me about that later. And this is a suite of analyses that I've run with different search parameters based on the algorithm requires some input from the user, and so I can talk about that if you're interested. But leads to dramatic improvements in model fit. Here's something kind of interesting that we can do. We can kind of smear these results together and look at the frequency of these shift events that we're detecting through time, controlling for the number of lineages as like a sliding window. And when we do this, we find that there are these really interesting kind of peaks in the frequency of these shift events that we're detecting. And I don't know if I'm allowed to do this in the context of like the research and congruence classes, but I'm doing it today. And when we do this, we find that there's like a perfect alignment between these peaks that we're detecting in the rate, in the number of shifts we're detecting, and these like climate shifts through the last 50 million years. So that seems really neat. I don't think that's a coincidence, but TBD. Okay, so just quickly going to the end. Excuse me. Yeah, I'm going to skip through this stuff. Okay. All right. So we can actually look at the the frequency of how often we have a rate increase and a rate decrease, and when we aggregate this across all the analyses that I've done, there's an overwhelming signal that that most of the rate shifts that we're finding are rate decreases rather than rate increases, and indeed the frequency of rate decreases is much higher than the frequency of rate increases, very similar to what Ian was just talking about. Indeed, the magnitude of a rate increase is larger than the magnitude of a rate decrease. So this is, I think, sort of consistent with a punctuated equilibrium type of model where you have cladogenetic shifts. This is a kind of a cladogenetic model, so I think it's sort of consistent with that kind of framework. Yeah, anyway, that's all I got. In summary, we're using computer vision to dramatically accelerate the rate at which we're generating phenotype data. We're measuring thousands of bird skeletons, and if you have a specimen collection that you'd like to work with us on, please get in touch. And I'm building a new multivariate framework for analyzing large phenotype data sets that I'm hoping will be useful to the community. So that it will eventually be in our package, so we'll get in touch. Get in touch if you'd like to use it. Okay, that's all I got. Thanks. I think I have like 1 minute or 2 minutes if anybody's got questions. Yeah.
Definitely.
Yeah.
Have many questions.
I'm sure you do. Yeah.
Two things come to mind. How do you guard against overfitting? And
Yeah.
also, how are you said hundreds of traits?
Theoretically, hundreds of dimensions, yeah. So it's using the underlying model fitting machinery from MVMorph in this framework called, Julian Clavel calls it, it's this MVGLS framework that uses penalized likelihood. So it uses a regularization approach to like shrink the variance covariance matrix. But I've only tested it with 10 to 15 dimensions, but theoretically it can work with hundreds of dimensions, and Julian has used it to study very high dimensional phenotypes. But he hasn't yet implemented a search type of algorithm quite like this. And so it's sort of TBD to see how well it will work for hundreds of dimensions, but it theoretically should work with that. It should run at least. It might be slow, but it should, yeah. 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 Okay, I think that's my time.
Hi, my name is Shelby, and I'm going to be talking to you a little bit about the work I've done this past year as a post-baccalaureate scholar in the Lagomarsino lab at Louisiana State University, looking at the evolution of Palychorea and chemical evolution within the clade. To start with a little bit of background, Palychorea is this super species-rich and diverse genus of tropical plants in the coffee family, or Rubiaceae. I've thrown up a handful of species up there to give you a sense for that diversity, but there's over 600 in total. One of the claims to fame for this group is they're known for producing a lot of really interesting and useful phytochemicals, many of which have a lot of implications for human life. Unfortunately, many contemporary studies over this group suffer from poor taxon sampling. Palicorea has a very broad neotropical distribution spanning from Central and South America, but most studies, including all currently published phylogenies, are limited exclusively to Central American taxa, which really doesn't cover the breadth of diversity that this clade has. So our goals for this project were first to expand the phylogeny to more accurately capture everything going on in this really cool group, and then to look at that through the lens of chemical evolution. We're going to start by working on the phylogeny. So this was made possible by our wonderful collaborators at the Missouri Botanical Garden, who lent us a couple hundred of their herbarium specimens. We began by using a modified sorbitol extraction method to pull the DNA from the herbarium specimens and then sent them off to Rapid Genomics to be sequenced. We were pulling target enrichment sequence data using a Rubiaceae-specific probe set designed by my lab mate, Lamone Ball. And from this, we were able to recover over 2,000 exons at over 1,000 different loci, which is a huge increase in the volume and quality of data available for this group, as prior studies were limited only to single locus sequences. We derived alignments in gene trees using the software tool HivePiper, and ultimately derived our species tree in Astral. And here is our phylogeny. This was building upon prior work done by my lab mate, Anna Bedoya. So we have 203 taxa total. We were responsible for adding 93 of those. It's the ones highlighted in green there. Here's a couple of them. And this work is still very much ongoing. We are currently awaiting sequence data for another 42 taxa. So hopefully in the coming months, this tree is going to get a lot bigger. So that was the phylogeny. We're going to switch gears a little bit and talk more about the chemical front. So primarily what we were interested in is this group called alkaloids. This is a very large and diverse group of nitrogen-containing phytochemicals. Here's some that you might recognize. If you don't, this is what you might know them from. As you can see, this group has a lot of relevance to us on pharmaceutical fronts, for cultural practices, and just general recreational uses. And they are known to occur within Palicorea. Actually, all of the taxa up on the screen right now are members of the coffee family, so these are all closely related to our study group. Their primary role within the plants themselves is to defend against pathogens and herbivory. We were also interested in looking at a couple other phytochemicals known to occur in Palicorea, including cyclotides, which are actually proteins characterized by a cyclical spine and 3 stabilizing disulfide bonds. There's ongoing research with some of our collaborators at the brain chemistry labs out in Wyoming, looking at how these can be used to treat aggressive forms of brain cancer, including glioblastomas. And within plants, some evidence has suggested that they actually work cooperatively with alkaloids to help prevent herbivory. It's really exciting that these are known to occur in Palicorea, as they are pretty difficult to screen for, and they've only been identified in a handful of plant groups. Palicorea just happens to be one of them. We also looked a little bit at flavonoids, which are just secondary metabolites with a polyphenic structure. These, for human purposes, have general anti-inflammatory and antioxidant properties, and their primary role within plants is flower pigmentation and to protect against UV. Unfortunately, screening for these, like I mentioned a little bit with cyclotides, is pretty difficult. It's expensive and requires a lot of resources that we didn't have access to for this project. So for our purposes, our main focus was to look for and extract alkaloids in the lab. And this was how we did it. This was a method that my lab mate Namwan and I adapted from the Stats Auto method and allowed us to use the herbarium specimens that we had on hand. So we began by taking the dried leaf tissue from the specimens. We ground them and suspended them in ethanol. We allowed 24 hours for the ethanol to extract phytochemicals from the tissue. We then poured off the extract and allowed it to evaporate. The tissue was then resuspended 2 more times, so the entire extraction process happens in 3 instances. And then at the end, what we're left with after all of the ethanol has evaporated off is this crude chemical extract. We then resuspended this in acetic acid, washed it with petroleum ether to help pull out lipids, chlorophyll, really anything that we're not interested in. And what we were left with is the alkaloid extract. So that's the part at the bottom there. So if the plant contained alkaloids, it would be in this extract. The next step was then to screen our samples. We screened each one against Dragendorf's, Wagner's, and Mayer's reagent. And what we were looking for, I don't know if you can see it in the picture up there, but how all three of those samples have a very opaque color and the white one has little dots in it. We're looking for the formation of a precipitate, so that is a positive result. The sample did have alkaloids in it. In total, we screened 167 species. We found 60 to be positive and 107 to be negative. And here are those results mapped onto our phylogeny from earlier. As you can see, it's a little bit sporadic. The presence doesn't really seem to fall into any immediately obvious patterns. One thing I do want to point out is that there are alkaloids present in the in group as well as the out groups, which indicate that the ability to produce alkaloids is likely the ancestral state. I do want to add the caveat that oftentimes plants will produce or will synthesize and store alkaloids in other parts, like such as leaves or the roots, and we were limited to only testing leaf tissue as that's what we had on hand. So just because we didn't identify alkaloids in some of these specimens doesn't mean that they're not in the plants at all. It's probable that at least a handful of them are able to produce them, but were not experiencing herbivory at the time that they were collected and therefore didn't have alkaloids in the leaves. We also wanted to look a little bit at some of the other chemical data that I mentioned earlier. So the cyclotides and the flavonoids, those are mapped out in the yellow and the green. Like I said, these are difficult to screen for, and that's not a problem that is exclusive to us. There was pretty limited data on which taxa are known to have these, but we were mainly looking for instances of co-occurrence. We found one instance of co-occurrence in Palicorea prunifolia, highlighted there with a little star, and 2 instances of co-occurrence with flavonoids in Palicorea crocea and patifolia. Crocea is actually interesting because indigenous groups in its native range were known to use it for its anti-inflammatory processes, so it's been the subject of a little more research just because it has a history of being relevant. And then again, I want to reiterate that it's very likely that there are more instances of co-occurrence due to the variable nature of alkaloid presence in the leaves. Hopefully future research can kind of flesh this out a little bit more and look at the plants more broadly than we were able to. Speaking of ongoing work, so on this front, like I mentioned earlier, we have 42 additional taxa that are in the process of being sequenced and will be added into the tree. This will result in a phylogeny with a grand total of about 245 taxa, which is roughly, it's between a third and a half of the clade. So in total, we're aiming to provide a more comprehensive picture of Palicorea that can then be built upon or compared for other methods, as this is, at least in my lab group, a really interesting group and something I feel like we could benefit from a lot. And more specifically, one of my lab mates that helped with the construction of the tree is working on fruit and flower morphology. She also plans to look at the nutritional compositions of the fruit, and she has some field trips lined up. Hopefully she'll be able to collect some relevant environmental data, and then we'll compare that to the results from this study. And then more broadly, the LAGO lab is taking a very holistic approach to understanding Rubiaceae, so the parent family, looking at this research, comparing it to pollination syndrome, fruit and flower morphology, altitude, climate, really any other potentially relevant factors. We want to get a good sense for what all is in this clade, or what all is in this family, how it's evolving, and the factors that are contributing to that. And then broadly, understanding patterns of chemical evolution can help inform more targeted studies in the future, especially for these groups that are so relevant to us. And with that, I want to give a huge thank you to all of the members of the LAGO lab, many of which are at this conference and had a hand in this project. I also want to thank my post-bac program, the LANYAP program out of LSU, all of my wonderful co-mentors and my cohort members, some of which are here today. Whoops, sorry. And our collaborators at the Missouri Botanical Garden and LSU's Shirley C. Tucker Herbarium. And with that, I will take any questions. Yes?
Is the genetic pathway for the production of alkaloid clear in this plant? And have you guys tried to look at the gene expression or the presence, absence of the component genes in the distribution?
Not yet. The genomic data was something we got back relatively recently, but there's been, I think most studies that have looked at alkaloid presence generally focus on biosynthetic pathways. I don't think there's been too much done in looking for specific genes, but that is an interesting, that's an interesting front to look at. We were considering kind of digging more into the alkaloids because looking more into the cyclotides on that front because they're proteins, but it could be expanded to alkaloids as well. Anybody else? Thank you.
All right. Thanks, everyone. Yeah, my name is Ray. I'm from the University of Texas at Arlington, and I'm here to talk about adaptive evolution and convergence. So, like every evolutionary biologist, I'm very interested in adaptive evolution and how we got the cool phenotypic diversity that we see in all animals today. And I've been particularly interested in how convergence can be used as evidence for that adaptive evolution, which has also been around since people have been thinking about evolution. But in the last couple of decades, it's got a bit more complicated than that. We found that convergence can just happen because, for random reasons, as evolution does. But it still, I think, has a powerful case to be show strong signals for adaptation, such as the classic example of African golden mole, a placental, and the marsupial mole, unrelated, but look exactly the same, at least on the outside. But I'm looking at Anseriformes. For those who don't know what Anseriformes are, they're ducks and relatives, geese, swans, and screamers. A benefit of working in ducks is I don't really have to introduce what they are to the people because everyone knows what a duck is. So why were you working on Anseriformes? Well, partly because literally everyone knows what a duck is. They are that successful. In every freshwater habitat in the world, you will find a duck. If you see fresh water somewhere, there will be a duck in it somewhere probably. They are that good. But they are also, the group is incredibly ecologically diverse. For something that's so morphologically conservative, and everyone can point at a duck or a goose and a swan and say, yeah, they're closely related. But they have this really wide range of ecological diversity, and dietary and habitat and all that, possibly one of the widest in birds. We have planktivorous filter feeders, we have herbivorous terrestrial species, we have diving species that eat crabs, we have species that eat fish. It's this really diverse group. And a lot of these ecological groups are convergent with one another. The most well-known example is what we call geese, and geese are just a hodgepodge of different lineages that all go on ground and eat grass, but they are not particularly closely related to each other. There have been at least 7 independent evolutions of this that in modern species. In fossil species, there's probably a few more. So this also occurs in diving. So there's a lot of independent evolutions of ecologies, which makes them, are they convergent morphologically as well? So that brings me to the first question. It's are they convergent morphologically as well as ecologically? And if there is convergence, is there convergence consistent across the body? Because a lot of these changes in ecology that I've been talking about also equal change in how they're getting their food. They're not just changing their diet, they're changing how they're getting their food, from going from swimming on the surface of the water to going on land or diving, which all require very different biomechanical properties and stuff. And finally, are these elements, these different bodily elements, evolving together or independently? Are they all integrated as one thing, or are they modular? Yeah, I'm very interested in all these types of questions. So before we go on, we made a tree. It's time dated, it's great, but I don't have time to talk about it today. If you want to know more, you can come up and ask me questions or talk to my PI/co-author, JC, who knows a lot more about it than I do. But yeah, using in all the analyses going forward. So I divided up Anseriformes into a bunch of ecological categories, starting with diet. These are just 5 different ecological dietary categories: straining on the surface, eating large invertebrates, herbivorous, mix, which is a bit of a generalist thing that most ecological groups have, and eating fish. And then I also want to look at different foraging methods in particular, terrestrial, surface, or diving, or wading. And then I kind of wanted to see how these interact, like these 9 unique ecotypes that exist, see if that is, how important factor that is compared to each thing individually. To quantify shape, did geometric morphometrics. We sampled 122 species, which is about 66% roughly of Anseriformes, depending on how many species you want to count in Anseriformes. And we landmarked the skull, femur, tibiotarsus, and tarsometatarssus to try and categorize as much of this morphology as possible. And of course, did the basic geometric morphometric things, did Procrustes superimposition, correcting for size, location, and rotation. We also wanted to kind of be as thorough as possible as testing for convergence, because pretty much every model to test for adaptive evolutionary convergence has some flaw in it that needs, that you just have to, it's there, you have to address it. So we try to get around this by just doing everything, chucking everything but the kitchen sink at it, which is using phylogenetic comparative methods, using C-score, which is a morphospace-based convergence thing, measuring changes in distance and moving through morphospace, and just plain old phylogenetic least squared analysis, right? So phylogenetic models we use to test: a Brownian motion model, a single peak Ornstein-Uhlenbeck model, where all the species converging to a single adaptive peak, 3 multi-peak OU models for diet, foraging, and this combined diet foraging group ecotype, and we determined best fit by sample size corrected AIC score. So let's get started with results. So the femur, foraging was by far the best fit model, significantly related to size, diet, and foraging, but interestingly not the combined ecotype. But you can see there's pretty good separation in the morphospace, which is surprising considering they're just femurs. And the biggest PC1, characterized by how curved the femur is compared to how straight the femur is, with divers having very curved femur, and terrestrial species having a straighter femur with a large trochanter. PC2 is also kind of associated with divers. Divers, it is the theme across this, divers have weird bones. So tibiotarsus. Also foraging best fit model, tibiotarsus was strange, and that was in our phylogenetic least squared analysis, it was only significantly related to foraging and not to size. For a limb bone, that just seems so bizarre to me, but it's not related to size, and not related to diet, and not related to any combination of the 3. Once again, in morphospace, divers are driving a lot of this variation. This is particularly by, I don't know if this cursor exists on here. Doesn't look like it, but at the top of the tibiotarsus, there's a big crest in divers that's there for stabilizing the leg, and divers have this very big crest. Everything else doesn't. When you go to tarsometatarssus, this is a very neat, nice, beautiful morphospace where they all divide up very nicely. Foraging, once again, the best fit model in the OUM, significantly related to every category that we measured. It also had the largest phylogenetic signal, still not a particularly strong one, but large phylogenetic signal. And once again, categorized by terrestrial to diving kind of spectrum, where long, skinny tarsometatarssus for waders and terrestrial species, and divers have very short, stout tarsometatarssus. With a slightly different positioning of the trochila, which are the little toe bits at the end. And finally, the skull is the only one that doesn't, foraging is not the best OU fit model, but this combined ecotype was. which makes sense. It's a skull. Diet is going to play a factor in what the shape is. But it is interesting that foraging is also such a significant factor in determining skull shape. The main differences in skull shape are bill length and how tall the skull is, and pretty much the facial orientation, whether it's very horizontally orientated, which are generally more predatory animals, or the very downward tilted faces. So, kind of like the overarching theme of what we got from the morphological measurements was foraging method seems to be the predominant factor determining phenotype in Anseriformes. And when we look at and map that onto that lovely tree we have, we can kind of see why. Foraging method changes all over the place in Anseriformes. It's not consistent for any long period of time across the tree. And there's very few groups that are only one thing, which is very cool. And when you map out, this is roughly averages of each of the elements. I like that you can kind of see a weird gradient between terrestrial and diving. Straight femur slightly gets curved, gets more curved. And you get, for all those bones, it kind of exists. But we can then kind of subcategorize within foraging these ecotype models of the skull. And we have this cool diversity. But we started this talking about convergence. So are these elements convergent? They show signals for adaptive evolution, but are they convergent? Because those aren't, the same thing. So using RC-based scores and all that, based on the categories, which were the best supported model for each element, all these elements are, we found a significant signal for convergence, which, as you can see, is a lot. Most of the hind limb stuff seems to be convergent, and at least 3 of the skull categories are. And 2 of those ecotypes have been convergent in every single element. I just wanted to highlight them because it looks great when you look them next to each other. The terrestrial herbivores, or what we call geese, really do look similar when you look at them. All very similar skulls, that straight femur, that long tarsus metatarsus, and the very short pointed beak. But also we found these diving invertebrates are convergent. These tall, robust skulls and these very strongly curving femurs and these very short tarsus metatarsus. These are animals like eiders and scoters up north, but also steamer ducks from down in South America and musk ducks from Australia. And all these animals also tend to at least partially inhabit coastal environments as well. So there's something in that. And finally, kind of all these elements seem to be adaptive, but do they covary with each other? Are they modular? Are they independent? And we found everything is covarying with everything. We expected with the hind limb that everything would covary since they all act as it's a functional one unit, but each of the hind limb elements also covary with the skull. All these bars here are scaled to effect size, the Z score, and you can see hind limb elements once again are all stronger, but they're not drastically stronger than covarying with the skull, which is really cool. So when we're looking at Anseriformes evolution, it might be a mistake to just look at one element because they seem to be very integrated across the skeleton. So just a big summary. Adaptive signal in Anseriformes associated with foraging behavior. There's convergence everywhere. 2 distinct, very convergent ecotypes. And there's a significant integration between all 4 elements. Thank you for listening, and thank you to all these people. And geese are also convergent in being mean. Thank you. Yeah.
Are diet, size, and foraging all coordinated? Because that synergy
Well, what threw that, looking at that off is, one, size was correlated, but it's very weak. It was a very weak signal, and also the fact that tibiotarsus just wasn't associated with size at all, and that if anything is very associated with the posture of like the bird. There's something interesting going on there, but that signal was hard to tangle out. It's not very clear when you looked at all the allometry plots and all that. There was rarely a significant interaction between like foraging and size. I think only in the femur and the skull that happened. So yeah, it's a strange one how size is interacting. Yep.
So there's strong phylogenetic signal in the morphology. Did you look at phylogenetic signal in morphology?
I haven't, but other previous people have. There is like a phylogenetic signal. Like there are like especially with the geese grade, there's like there's a couple of like big groups. But if you look at the tree, there's also throughout it very often just like single species within an entire lineage. Like, oh, I'm diving now for some reason, or so there doesn't seem to be a strong phylogenetic signal with ecology. There is one, but it's weaker than would be expected.
Because I'd be interested in the discordance between the phylogenetic signal in the morphology and the ecology. If one is stronger than the other.
Yeah.
Which one's stronger?
Yeah, that's something to look at. That yeah, I haven't looked at it, but that is something to look at. Yeah, thank you.